Have you heard the term triple planetary crisis before? The three topics we deal with in parallel are climate change, biodiversity loss, and pollution. Today's conversation looks deeper into the topic of biodiversity loss. Our planet currently goes through its sixth mass extinction period, and today's guest dedicates her career to the question, is the species really extinct? Christina Biggs is a marine ecologist who takes us with her into the deep sea, to the Galapagos Islands, and into the ecosystem that everyone can explore without getting wet feet, the aquarium. She dedicates all these trips to finding species that scientists believe are extinct or at a high risk of extinction. We learn what it takes to find something we believe is lost and get a glimpse into how ecosystems work and why even a tiny centipede can change the air we breathe. Christina has a fascinating talent to speak in images, and she also conveys hope in a dire situation. This conversation is not all doom and gloom, so do stick around to learn why there's still reason for hope. This is Where the Ocean Meets Christina Biggs. Hi, Christina. It's a pleasure to have you here today.
Hi, Joel. Thanks for inviting me.
Your LinkedIn says that you are a conservation ecologist. What is that?
So I basically take ecological research data and apply it to situations that require intervention to help ecosystems and species thrive and survive. And a lot of it is restoration work as well.
Mhm. What does a typical day in your life looks like? Because I haven't heard of that job before. So it would be great if you can give us a bit of an insight into what your day to day is.
Okay. I think I have two separate day-to-days. I have the days that I sit at my computer and look through data, whether it is IUCN red list data, spreadsheets. I have a lot of meetings talking to other people to identify problems and work out solutions and strategies. And then my other day, which would be fieldwork days, is typically traveling to, for me, it's remote places. I work in areas where it's under-resourced and understudied, so places where there are high levels of species and endemic species that are only found in those places that are typically pretty difficult to get to. So those days look like camping or ship-bound activities. I do a lot of marine work, so days on boats, and that's usually collecting samples, whether that be water samples for eDNA or photos on camera traps. Observations of species, and then putting that all in, recording that so that we can use it when we get back to our desks and go back to that desk work day.
Interesting. And is there actually a difference between a marine biologist and an ecologist?
It's slight. I think the two terms are often used interchangeably. I think marine biology tends to focus specifically on, you know, I tend to think marine biologists themselves have one area of interest, say octopuses, and they're focused on studying everything and all that you can know about, say, a particular species of octopus. Ecologists tend to look more at holistic systems and how the species within them interact, and again, what needs to be done to make sure that those systems are functioning properly and healthily. So, yeah.
Interesting. Uh, And I think for most average Joes, the way how to look at the ecosystem of the ocean is also where you started your work, is basically an aquarium, right? Or maybe if you're on vacation, you can go snorkeling and you see a bit of a reef or a little bit of beach. But if we want to see ecosystems, we go into aquariums. And my understanding is that this is where your career started. Is it, or when is it ethically correct, would you say, to visit an aquarium? What do I need to look out for if I want to make sure that I support the right thing?
Yeah, accreditation is a really good place to start. Here in the United States, we have the Association of Zoos and Aquariums, AZA, and that process to get that accreditation is very robust. The criteria you have to meet, it's a high level of standards, and most of the aquariums in the U.S., hit that standard. And there's also the World Association of Zoos and Aquariums, WAZA. So, that's the body that oversees other aquariums worldwide. So that would be my starting place, is to make sure that they're accredited institutions that follow high levels of animal care and ethical practices.
If I have an aquarium at home, right, I sometimes ask myself, can these fish be happy in this little, like, I don't have an aquarium, but when I see that and I see this little box, I sometimes think it's like, can these fish be happy? I mean, probably we can't measure happiness in a fish, but how does it work for them? Do you think they are content or?
Yeah, I mean, happiness and content, like you said, they're primarily human associated traits, but obviously there are things that we can look for: stress signals, diseases, bored habit patterns of swimming, things like that. And so there have been years and years of study to show what what those signs of stress look like, and ways to mitigate that. And I mean, one of the benchmarks is, if there's breeding going on, that's a level of comfort that animals will exhibit once they're, if you want to say, content or happy or comfortable. So you can you could maybe use that as your high level benchmark for fish happiness.
Okay, yeah, that makes sense. And uh, I mean, now you're not in an aquarium anymore. You are out in the ocean. You go to places like the Galapagos, but I want to stay on the aquariums for a little bit. What do you think is their role in understanding ecosystems better? Why do you think they are important?
I mean, I think they're pivotally important. Even though I'm not in that sector anymore, it's still a group that I interact with almost daily. And I think, in this time, this extinction crisis that we're in, the knowledge that is being accumulated within aquariums about animal behavior, and like I just talked about, conservation, breeding, all of these things are applicable to the field studies. Once we know them, when we have them in a controlled situation, in a lab situation, and can suss out what are those needs and those important things, then we can apply that directly to the field. Also, I mean, these days, biobanks, I mean, having genetics outside of places where there may not be any habitat left for these species to exist, and the only place that they can exist, if at all, is going to be in aquariums, with the hope that at some point they can be put back or rewilded. Um, and then there's just the sheer number of people that aquariums and zoos reach every year, for conservation messaging. And I if we want to go way far back to my aquarium days, I mean, my parents took me to SeaWorld when I was 6 years old, and I was hooked, and then ended up pursuing a career many years later in the conservation field. So, as far as inspiration, education, a lot of fundraising happens. It's a great place to get it. It's hard to raise money for field research, and a lot of the conservation initiatives now start at zoos and aquariums worldwide.
Hmm. I mean, this is part of the reason why I do this podcast, because I want to ignite this at least interest in people for the oceans and the importance, because I think we need to protect this ecosystem. And I'm happy that you got hooked, because you're doing so much interesting work now. You mentioned um, that these, or that we can learn from the aquarium for what is then applied in the real world. Are there any examples that you could highlight where this transfer worked? Where
Yeah, I'm thinking of, I'm not going to name the exact species, but there was a project in the Western United States where there was a really low population of fish, and they kept trying to put the fish back. They would stock them in captivity and then put them in the wild, and the populations just kept ending up depleted. And so they did a series of studies in the aquarium and learned that although these fish they thought were habitat or um, they lived in these rivers that had like a high flow rate, like, raging rivers. When they were in the larval stage, they needed like quiet little ponds. And so in the rivers themselves, of course, all rivers have like little blockages and side channels, and that's where the larvae were going, but they didn't know that. And so they were putting these larvae into these raging rivers, and they weren't surviving, and they couldn't figure out why. And then they realized that they just needed to place them in a slightly different section of the river. And then when they grew large enough, they would go and migrate into the river and live their adult lives and populate. So doing those studies in the aquarium allowed them to figure out like, hey, it needs this situation, not that situation at this stage of its life. And therefore we can be successful and help this population grow outside of the aquarium.
Mm. It sounds like a rough start to life if you just put into a raging river. um, if you think back to these days in the aquarium yourself, now we learned that part is data in your day-to-day job today, and part is being out in expeditions. What did you do in the aquarium?
Um, just a wide variety of tasks. Part of it is exhibitry, so making sure that the displays are as accurate as possible for the guests and are visually pleasing and have happy healthy animals in them. So part of it was exhibit maintenance, design, and animal care. Part of it was also that research that we were just talking about. I mean, I used octopuses as an example earlier because I worked mainly with cephalopods, and so we did a lot of breeding experiments with those octopuses and cuttlefish and nautilus. So I did a lot of lab work as well. Diving, obviously, diving programs, which is one of the more fun parts of the job as well. And then educational programming and speaking, and did some media to help talk about these species and make other people fascinated about them.
Mm. There's so many ways we could go from here, because I saw that you also studied journalism, if I'm not completely wrong.
I I did. I studied writing, yes.
Yeah, and I think the communication part is so important, and also reaching audiences that are not interested yet, and how to excite them about what's going on. And you dropped the term extinction crisis earlier, and I think this is something that might interest more people, because suddenly it becomes personal, right? Because we are somehow interconnected. Can you describe what you mean by extinction crisis?
Yeah, I can definitely tell you it touches my personal life. I have family members who say, why do you travel half a world away and muck through mud up to your eyeballs to go try to find a lemur species that hasn't been seen in 20 years? Why do you care about that? Like why should I care about that? And so I often use this interconnected, the world is interconnected, and that, my family in the Midwest, the air that they breathe isn't necessarily produced by the tree that's in their backyard. It could be produced by a tree in Madagascar, end up, in the jet stream and moved all the way around the world until you're pulling it into your lungs. And that tree can't grow unless the lemur eats the seeds, poops the seeds so that it can germinate into the tree that produces the oxygen that I'm breathing today. So, it isn't the lines that we've made up. While they may be important for different reasons, they're all imaginary. It's one world. All of the resources we have here, all of the clean water that we need to drink and the air we need to breathe, comes from every single place.
Mm. And what is this extinction crisis exactly? Like, what are we facing there right now?
Yeah, so the data clearly and unequivocally shows that we have entered the sixth mass extinction. So there are the ones that are famous that people know about. The extinction of the dinosaurs was a mass extinction. We are now in a mass extinction in that we're losing hundreds of species every day, some of which we haven't even discovered yet, that are dying due to specifically human pressures, human caused pressures, climate change. Um, Habitat destruction is probably the biggest thing, human wildlife conflict. So all of those things are driving, important keystone species to the brink of not being able to survive. And we see it in the oceans alone. I think fish stocks are down 70% from healthy levels. Just staggering numbers, which when you talked about storytelling and the writing component and things like that, I mean, I'm I'm immersed in this, so I hear these stories every day, and I'm panicked every day thinking, "How can we go on? How is this world gonna survive?" But I don't think, you know, for the average person, you talked about the average Joe, I don't think, that news gets disseminated. We're all looking at the things that are important to us, and, if you're in the financial world, that may be stocks, or you know what I mean? Like, everyone's living their daily lives, and they are surrounded by the people that are like them and influence them. So they may not be getting these stats and this information about this mass extinction. In fact, I think most people probably haven't even heard that terminology. So yeah, it's important to tell these stories.
Do you meet people that deny these facts, like of the sixth mass extinction? Because I know
Um,
there's climate change deniers, but the numbers are so clear of species lost, right?
yeah, the data is factual and doesn't lie. But of course, I do come across people like that, and um, it's difficult, right? I try to keep an open mind when I listen to people, but you know, I think the problem these days, the internet is obviously a great gift to all of us in the fact that we get our information so quickly and immediately, but then the opportunity to have false information put around and to make people think that they're experts. Like, I'm a marine ecologist. I'm not a climate change scientist. So when those people who have studied for decades and have done studies for decades, and use methods that have been tried and true scientific methods, I believe them because I know how hard it is to do my job and what I've had to do to get there. And I believe that they've done the same thing, which gives them an expertise and a voice that's, authoritative and believable. And so I believe climate change scientists. In fact, I don't even think it's believing. It's not Santa Claus. It's
No,
science. And as a scientist,
exactly.
I
Yeah.
believe science. So
Yeah. How does your family react, for example, when you explain how these things are interconnected? Do you feel like there's an open ear for it? We also don't have to talk about your family, but in the wider
no,
space.
my well, I'm lucky. My family's lovely, so they ask out of genuine curiosity. I know that some people who wanna deny this act out of antagonism. They wanna start a fight with you, and they wanna tell you you're wrong and, again, pull all their false facts off the internet to throw in your face. My family asks because they wanna know, "Why do I do this?" Like, it's important to me. Should it be important to them as well? So, they openly listen, which is lovely.
No. Yeah, then let's try and get some more understanding. And I find the topic that you're working on, this extinction, super interesting. And I don't even know how to get started on it. How did you get into this extinction in the first place?
Well, the program that I ran was called the Lost Species Program. And so basically, it's a list of species based on a definition of non documentation. So if a species hasn't been seen in the wild in over 10 years, then it would fall onto this lost species list. Um, it's an interesting list because it could be species that are in true crisis and at risk of extinction. It could also be species that are in remote places that people don't go visit. It could be that they're not, lovely, cuddly panda bears. They're centipedes, and people, you know, less research goes into invertebrates than it does into mammals. So there were a lot of reasons why species would show up on this list. And my job was to try to figure out which ones did fall into that category of this is a true problem. And a lot of those we saw on islands, places where species are bordered. They can't move. If their trees all burn down, there's no place to go, to find new trees. So a lot of these were focused on islands. So my job was to identify which species were at risk of extinction. So that's how I jumped in and tried to become as knowledgeable as I could on the subject.
How does it work? How do you identify this? Because maybe I'm naive, but I can't imagine that there's a lot of people out there who constantly monitors, like, when did I last see this invertebrate? When did we discover this? How did that list come to be?
Well, a lot of things in the world of conservation start with the IUCN, right? International Union for the Conservation of Nature. So they have a list. A lot of people have heard the term red list. The red list is actually just a giant database of information that's been provided by scientists, by a network of something like 10,000 volunteer scientists, and they assess um, the stability of species. It's a little bit skewed because, again, a lot of research in the past has gone specifically into larger, more charismatic animals, and so things like centipedes and flies maybe haven't gotten as much attention. But there are specialist groups for all these species, and they do these assessments. Hopefully, every 10 years. It's hard. It's a a huge number. There are a million species on Earth, so they do these assessments and they put this information online. I access that information and I can look for certain terms like not seen in 10 years or only known from holotype, which means it's only known from the first time it was ever seen and described in a scientific paper. So mostly my information all started from IUCN.
Hmm. Is there any prominent animal that comes to mind that's on the list? And to contrast that, any particular animal that you're specifically worried about?
Oh, there's pretty much 4,500 species that
Oof,
I'm
okay.
worried about. I mean, again, climate change and the extinction crisis, those numbers, those statistics I gave earlier, 70% declines, 90% declines, habitat loss. Um, there are literally thousands of species that are at true and real risk of extinction. So as part of that lost species program, we curated a list of the top 25. It wasn't necessarily the top 25 that were in immediate crisis. It was a list that sort of showed how vast the problem is. It was all geographies, so it spanned from Indonesia to Kazakhstan to South America to the Arctic, so that it shows that these species that are in trouble are in all habitats, all regions, and all classifications. So there's a shark on the list. There's a lemur on the list. There was a spider on the list. And so, it's a vast problem. To say that I'm worried about maybe necessarily any one thing, I'm worried about a lot of things.
And for someone that doesn't understand much about ecosystems, for example, if a specific fly goes extinct, why is that such a problem? Can we say it's like, oh, maybe that was just evolution and survival of the fittest? It's not a fit one. Why is it so critical? Or how do these systems
Yeah.
work?
So if you look at the data science that goes back, thousands of years into the cycles of extinction, yes, there's evolution causes certain things that are not functioning properly or not integrated or don't have the support to die out. That happens naturally. But that attrition is very, very small compared to the things that adapt and thrive. That's why evolution is so incredibly successful, is because it adapts. It's adaptive, and it forces species to become dominant, prominent. So if you look at the numbers now and what's going extinct, those numbers are exponentially higher to the point where it's not natural evolution. It's an external force that's causing it. And that force, of course, is humans and human activity. So yes, for people who claim like, oh, well, things have gone extinct all throughout history, that's true. But you have to look at, the last 10,000 years, even the mammoths went extinct because humans became hunters and, hunted them to extinction. So the reason for extinction, while it does happen, definitely the data says it has grown exponentially due to human activity. To answer the second part of your question, why does a fly matter? Um, you know, Ecosystems are just that. They're these contained borders that have all of the things in them that work together to create the ecosystem that functions the way it does because of all the moving parts. I mean, it's just like your car. Why do you have tires? you could still call it a car and it wouldn't have tires, but it wouldn't drive. So, just like any kind of mechanical thing, all the parts come together to make it 100% functioning.
No. So even if there's a centipede that goes extinct, that could lead to ecosystem collapse.
Yeah, I mean, to give that example, centipedes burrow, they aerate the soil. Seeds need air within the soil to grow. Without that air, the seeds may not be able to germinate, the trees may not be able to grow. Then there's no habitat for birds to nest, and there's no fruits for primates to eat, all of those things. So it may seem like it's just a centipede, but it's an engineer. Everything that works in an ecosystem is essentially an engineer. And so for centipedes, it's aerating the soil. If the soil becomes compact and nothing can grow in it, it's barren.
Mm. Which in its last instance is also bad for us humans, right? So this connection is so important, and I wish more people would look at it this way. Um, I remember reading a quote. I think it comes from Darwin himself, who said he can't even really enjoy a walk in the forest because all he sees is nature killing itself. How is it for you if you go out there and explore these ecosystems and see the food chains working? Because I like your description much better, right? It's like the centipede is helping with the soil and helps grow plants. So that is a very supportive description, where the one from Darwin is quite like everything dies and everything fights. It's
Uh, while I understand what he's saying, it's I do enjoy nature, but I mean, there have been times that I've been out on boats. I saw orcas attack and kill a porpoise, and it was brutal. I mean, it is. I've seen the wildebeest crossing in Tanzania, and the crocodiles grabbing, a baby wildebeest and pulling it under, and all of those things are horrific when you think about them on that level. But in the circle of life, I mean, I can appreciate the circle of life that everything eats and feeds. And I hope that I just saw this is a little bit of a divergence, but I just saw a mushroom coffin, right? A mushroom, it's a fungi based biodegradable coffin. And basically, within 45 days, it decomposes. And then anything that grows above it basically is you when,
that's beautiful.
after you
So
die. And it's a beautiful, like, for me, I've said I want a sea burial. Like, I want to go be eaten by the sharks. I love it.
uh,
I love the idea of that. Like, we're all sort of part of this circle of life. So, I don't quite see the the constant war that Darwin sees. I'm fascinated. I will say my family goes crazy because every time we go for a walk, I am not walking. I'm stopping and, oh, look at this. There's a banana slug, or oh my gosh, that's a blue jay, or I see the wonder in it. But yeah, I get Darwin's point for sure.
yeah. No, but I'm happy that I now got your point of view. I like that a lot more. Um, Coming back to the ecosystems, and I mean specifically the ocean. Now you have this list, and let's say there's a species on there, and you're trying to discover whether or not it's still out there. I think it's harder to do that than finding a needle in a haystack. How do you approach something like this, especially if it's these tiny little species or anything at all? Because basically you're a detective.
Yeah, that's exactly what it is. I mean, it's the most puzzling and fascinating investigative work that I certainly have been a part of. It's complicated, but there are steps. I mean, obviously, you start with that description that I talked about, like the first academic sighting. Where was this found? And if it's something that was once abundant, obviously you have lots of data as to where it was seen. IUCN keeps these species ranges, so I can look and say, okay, hundreds of them were seen in this one little area. Obviously, that's where you're going to start. You're going to start where it was first seen or where it was last seen. You're going to bring in experts. I am an expert on octopuses. There are a couple octopuses on the list, but for the most part, I'm going to go find an entomologist or an ornithologist, and particularly someone who has an interest in that family or genus of species. So they know their traits. They know what they want to eat. They know where they make their nests. They know how and when they breed, how and when they sleep. All of those things, so that you make sure you're looking at the right time, because I have seen people do searches And they're like, I looked thoroughly in this entire area and I didn't find it. And it's like, well, did you look during the daytime? Because they're nocturnal, and you're gonna need to go out at night, or simple things like that. So you make sure that you're looking at their natural history and you're actually looking for something that could be found within the parameters of how you're searching for it. Um,
Mhm.
these days, we use a lot of technology. There's great emerging technology. I'm working very closely with eDNA scientists that I find to be a really valuable tool. It maybe doesn't help you cite it, but if you can find genetic confirmation that it is there, then again, you can then apply other tools to actually get a documentation.
I keep hearing the term eDNA. What is it? How does it work?
Yeah, it's emerging and it's becoming widely used. So eDNA is environmental DNA. It's all of the genetic material that gets left behind in an environment. So it's skin cells, it's feces, it's mucus, it's anything that's shed. We shed our hair, our skin, our fingernails, all of those things. Animals leave that behind too, and it persists, whether it's in water or soil, in the air, on tree canopies, those types of things. So we can collect that, we can filter out those little genetic particles, and we can send them to a laboratory. And through those sequences, the DNA that we all have, they can make a snapshot essentially of what is in that environment just through um, discarded DNA essentially.
Mm. I would imagine, like, if I pull a liter of seawater, that there must be thousands of species and eDNA in there to be discovered. Is that the case?
It's definitely the case. It's absolutely and utterly fascinating. I like to combine technologies. I like to have taxonomists who can make visual identifications alongside the eDNA, because both of them pick up things that the others missed. I mean, it's really hard when you're diving in the ocean. You've got a little slate. You're fighting currents, and you're trying to see, and things are darting off. So you make a list of species that you see, and then um, you bring filters with, and you're filtering the water that you're swimming through. So then you can analyze it later and say, oh, there were 50 things that I didn't see that were there. Next time I'm going to look for those. So
So
yeah.
it works in both ways. You can say, okay, now we have X amount of eDNA individual samples collected. These are the following species that are around, but I can also go in and say, it's like, is this specific DNA present in an area?
Yeah, we did a study in northern Madagascar, which was just published, and we did just that. We had eDNA and we had taxonomists in a wide variety of species. So um, we studied birds, fishes, mammals, reptiles, amphibians, and then also collected water from a variety of different sources. There were rivers and tributaries, but we also just found like standing puddles. Anywhere we could find enough water to put through our filters, we did. And then we compared the two lists against each other and with each other. And so there were eDNA outperformed the visual scientists by a little bit in every category, but there were things that the taxonomists picked up that the eDNA didn't. So we found that together combined, um, those two methods produced a bigger and more comprehensive, a better list of biodiversity in that area.
If or how long is there a spoil date on eDNA? Or could it be that by chance you're catching a piece of eDNA from an animal that was there 50,000 years ago?
Oh, well, those are amazing technical questions. To give you the high level answer, there is a study of ancient DNA and there's a study of local immediate DNA. So there are two different ways of looking. So if you're metabarcoding for DNA right now today, you won't pick up that ancient DNA. You have to use a different technique and a different way of looking for it. So while both persist, you won't get them crossed in your studies. You won't all of a sudden pull up and say, "Oh my gosh, there's a saber-toothed tiger here," because clearly there isn't.
That would
So
be hilarious.
yeah, wouldn't that be
they,
great?
they just moved underwater. They're now in the reefs.
Although a funny story with that is that a lot of times when they're putting in, say, like offshore wind technology or things like that, they do eDNA studies to make sure that they're not causing more damage. And I heard of one study where they kept picking up whale data in a place where these whales weren't known. And it turns out it was a shift. There was like a climate change shift. And so the whales that they didn't even know had sort of moved further north were picked up. And they did the study over and over again, saying, like, this is impossible. These can't be here. And then all of a sudden, a scientist was doing a flyover and was like, oh, well, there is one. So I guess
Yeah,
they are here.
that's crazy. And so I understand the part of the eDNA. So with a water sample, you can basically explore the tenets of that ecosystem. And then the other issue is that the ocean is not a very accessible ecosystem, at least starting at a certain depth. And what tools do you use then underwater? Because obviously I can go snorkeling, diving.
Yeah. Um, Also a complicated question, but nowadays, for when I worked at the Monterey Bay Aquarium and Research Institute, we had ocean-going vessels with extraordinarily tech-forward ROVs, remote operated vehicles with cameras and detection, like it would detect pH and water flow and temperature, all of those things. So highly detectable instruments. We could collect eDNA. We could collect organisms if we wanted to. We were mapping. It's using lidar and it's mapping the seafloor. So there is equipment that allows us to go now miles deep into the ocean. Obviously, that is millions in millions of dollars. And there are very few of them. I think there are only 6 or 8 organizations that have that kind of extremely high tech equipment. So so they are mapping, and they're like Schmidt Ocean Institute, for instance, has sectioned off the oceans into seven different geographic areas, and over the next 7 years, they have plans, um, to research each one of those areas in depth. So that work is ongoing, but it's limited to who can afford to do that. So as far as affordability and making this work, the survey work more effective, finding solutions that allow, especially local communities, indigenous peoples, people who are the stakeholders and who care about these lands and steward these lands and have for generations, to make that more accessible. We're developing tools, low tech, easy to use tools that can provide high impact
Hmm.
information, like the eDNA. In the Galapagos, we just piloted a diver tow, snorkel tow system that can filter, hundreds of liters of water at a time. And the other thing is, really, we have to invest in capacity within the countries to be able to process this information at their universities, in country labs, rather than shipping it to, right now the UK and the US have great laboratories to do this, but that's expensive and difficult, and then takes the information away from where it belongs and who needs it the most.
Hmm. I, I just had a question again on the eDNA that keeps floating around in my mind, and maybe the answer is too difficult. But sometimes I sit in front of my computer. I now have AirPods on, and it's crazy to me that Bluetooth just works and that it transmits the signals and so on. Is there an easy way to explain how to decode DNA or eDNA? What are the steps after I pulled that water sample? Sorry, I'm jumping between topics,
No,
but
it's a good question, because I'm not a molecular scientist, right? So that
yeah.
was the first question I had when I said, "Well, I want to utilize this. I want this to work, and I want to know how this data works." And I jumped into it, and then finally I was like, "I don't even realize what they're doing with it. Like, I just send it away somewhere and it comes back, but I should really know." So someone did write a paper, and it basically was like eDNA steps for the layperson. And really, what happens is you send your sample, you pull the DNA out, you put it through a filter, and then you preserve that filter and send it to a lab. Once it's at the lab, they dissolve the filter and then they use chemicals to wash out all the genetic material, and it ends up in like a little pellet. And then that pellet has all the eDNA strands in there, but they're too weak to see. So they make copies of it. So they basically through it's the same test that they use to test for COVID. You
Mm.
use the PCR machine and you replicate that DNA, those DNA strands many, many, many times, so that when you put it through the processing, the computer can say, "Oh, I can see that now. It's enough strands that I can see that is a strand of DNA." And then the computer spits out all of and it's all in A's and T's and C's and G's, and you'll look at it and it just looks like someone sat on the keyboard for too long and it spit out a random assortment. Um, but then you put it through the most important thing, and the thing that's limiting us the most right now is that you need to have you need to have the DNA sequence from the species to be able to compare those two to say, oh, it's that one. And so a lot of things have been DNA sequenced. Obviously, humans have been now,
Yeah.
but thousands of them have. But then thousands of them haven't, especially in these remote areas where you barely even know something exists. How could it possibly have a sequence in a database? So yeah, it's kind of chicken and
So
egg a little bit.
is it then that, for example, in this database, I could have an eDNA sample where I don't know what species is behind it?
Definitely. In that Madagascar study that I talked about, we had many, many, many that weren't. But almost everything is sequenced to family or genus level in the tree, so you can get it close. And then, for me, I still find it valuable to know. You can say, I have these 50 species that I don't know what they are, but I know that they're amphibians. And I
Mm.
know in this forest, we've only described 13 amphibians, which means if there are 50 individual amphibians here that I have different DNA sequences for, there are 37 then that I haven't seen yet with my eyes, and we need to know. I mean, you can't protect what you don't know. So basically, I mean, I guess I'm saying knowing what you don't know will help you figure out, like, hey, this area is important. There's a lot more amphibians here than we thought based on just visual surveys. So we need to take a deeper look and we need to figure out what these species are.
It's fascinating. It's again one of those cases where I just think humans can be so smart and so ingenious. We should be able to solve some of the problems. Um,
It's good.
going,
It's what keeps me optimistic for sure.
yeah, exactly. Me as well. Going back into the ocean, we briefly touched on the deep sea, right, and on the robots that we can now send underwater. Why is it even important to understand the deep sea? So far, I think people think there's not much going on, like compared to the coral reef ecosystem where you have so much life. The deep sea is the desert.
Absolutely could be further from the truth. And it's, I mean, but it's a common conclusion to come to. I mean, you can't see what's down there. There's no light. It's miles away. The pictures that we have are with artificial light and cameras in current situations with lots of debris. So it does look upon first glance like, oh, it's just, maybe a couple weird fish and some hydrothermal vents and maybe some crabs and things. But when you get down there, I mean, the amount of life is staggering, and the amount of undiscovered life. I mean, every year, because of these 6 or 8 organizations that are able to go down there and take a look, I mean, they're finding vast deep sea coral reefs. So beautiful things like you see at the surface, but 3 miles down, and different species, and with different life that they're supporting. So it's absolutely fascinating, and probably the area where in the coming years we'll make the most new discoveries as far as species, and there will be thousands of them. Um, why it's important? I mean, right now, one of the biggest things, obviously, we're looking for alternative energies, and I own an electric vehicle which has, metals in it that are needed for the batteries and things. And so, as that technology takes off, we're looking for new sources, and deep sea mining is one of the biggest sources of metals, and it's not regulated. On land, when you go, like I was talking about, like offshore wind energy and things, when you go to apply for permits to do that kind of expansion, there are ecological studies that go with it, and they can say, no, there's an endangered, you know, heron there, and so
Mm,
that's their habitat. You can't build there because of this. But with the deep sea, there is no regulatory. There's very little regulatory. It's vast. It's hard. Who owns it? Who owns the ocean? Off the coast of there are rules on how far out to sea nations own things, and so there's a lot of unregulated space. And therefore, it's kind of like the wild, wild west. Anyone can do anything. And so
mm,
you have all this deep sea exploration for metal mining, and it's destroying entire habitats that haven't even been explored yet.
mm,
And so that's one of the reasons it's important to know about these things. Again, everything is worth saving. Um, everything acts within its environment. And, frankly, the deep sea is where a lot of our carbon sinks. It's where we're keeping the climate for the rest of the world. The oceans regulate everything on land. It's 3/4 of the earth. So the oceans regulate everything that we do in our homes, far away from the ocean. The ocean is actually kind of ruling our lives. So it's important.
yeah. By the way, on the topic of deep sea mining and material security and all that, there's a fantastic book called Material World. I forgot the author's name, but the book itself is great. Um, but if I connect this back to something that we discussed earlier in the conversation, right? It's like even the loss of a fly can lead to an ecosystem changing, at least, maybe even to collapse. How far do we already understand the connection between what's happening in the deep sea with what's feeding us, what's oxygenating us? I'm not even
Mhm.
sure if that's a word, but um, because I heard this fact that we know less about the ocean than about space.
Yeah.
Um,
Yeah, I think we're really at, the tip of the iceberg. I think as we've understood, I think someone told me we're about 40 years behind in ocean science than we are in terrestrial science. So we know that it is important, and we know that research needs to be done. I think it just is just starting to be done. But that said, I mean, we understand the carbon cycle. We understand carbon storage. We know that there's a sink. We understand the zones of the ocean and, the role each one plays, the pelagic zone, the mesozone, all of those things. And we are starting to develop technologies to be able to um, do this more at scale. And even the Galapagos project that you mentioned earlier, one of the things that was really unique and very cool for me in that project is that we had a group of rebreather divers, technical divers. So we did a comparison, which has never been done, which is my dive team. I'm not a technical diver, so I dive within normal recreational limits. So my dive team was doing eDNA sampling at 5 to 15 meters, and then the rebreather team was going down to 60 to 75 meters, and yeah, and staying down there for 4 or 5 hours as regulations mandate for them to be able to decompress. So they drop down to the bottom, and then they come up at increments over the course of 4 or 5 hours. But within that, they were able to do an amazing amount of data gathering. They did have the eDNA tows. They also were collecting video transects, but almost everything they're seeing there, because it hasn't been studied. Like, we have these ROVs that go down to 3 miles, and then we have obviously surface divers who've been doing this work forever. That mesozone, where there still is light and there still is a huge amount of life that's different from what's at the surface. And so almost all of that is undiscovered. And, they came back up and said, I'm pretty sure I saw 3 new fish that have never been described. And, they rediscovered this coral that hadn't been seen, which is a whole nother story. And so, we have this technology, we have these people, they have nerves of steel. I have to say, I admire them so much. Um, doing this work to help us go into places that we haven't been able to go into previously.
I would like to touch on that And I have a couple of technical questions first. What is a rebreather?
Mhm. So when you dive, right, I think a lot of people have heard of the bends, right? You end up with nitrogen in your system. It collects from the pressure. And so you have to let that get out of your system before you come back to the surface, or it causes bubbles and you get really sick. And um, so technical divers, because they're down there so long, they accumulate a lot more nitrogen. And so they have this complex technology, which I wish they could speak to it because I'm afraid I might blunder it. But they have a mix of gases. Instead of just the usual gases that are used in lower depths, they have many gases. And so they have different canisters, and they have the technology that mixes those gases dependent on where they are in the water column. So, they'll have a higher mix of helium lower in the water so that they are not accumulating that nitrogen. And then as they start to come up, they change those mixes, and they have complicated computer systems that are part of their dive watches that attach to all of these canisters and all of this equipment that allows them to, um, based on where they are, remix gases in real time on the fly to adjust.
That
Um,
is so crazy.
yeah, and I watch them test their equipment, which is utter fascination. As they're getting ready for their pre-dive check is nothing like
It's
my
not the
pre-dive
standard
check.
buddy check.
Yeah, it's not your standard buddy
Yeah,
check.
you
And
have your goggles.
yeah, they have scopes that look into the tubing, that they're like looking through scopes and watching valves open and close and things. And it was mind-boggling to me.
They must really love their job to go to
They
that
do.
risk.
Yes, they're good at it.
Do they hate people, and that's why they want to stay in the ocean for 5 hours without? No. Second question is, you mentioned the name mezzanine.
Yeah, the mesophotic zone. Yep.
What is that?
So mesophotic zone is it's like where the light and depths change. It's typically between like 100 and 300 to 450 meters. So again, below recreational diving limits, but not open. Um, you get to a point, obviously, as you're descending in the water column where there's no structure around you. It's just big open ocean. That's like the pelagic zone. Below that is the twilight zone, where light ceases to exist, and then you find all those creatures with those crazy adaptations, like no eyeballs and glowing appendages and things like that. So the mesozone, I don't want to say regular creatures like normal fish, but it's a different mixture of fish because they're living deeper. It's less light. You don't have it's like you don't have kelp forests. You don't have those kinds of like algae. It's a different composition, a different food web. Obviously different temperatures, different lights, but it's just below the reef zone,
Mm.
but before the pelagic zone.
Okay. And you already mentioned you can find some fish there, and you discovered a coral. What was so special about discovering that coral there?
Yeah, so the coral was on that lost species list that I told you about in the 25, the top 25 list. That coral was on that list. It was last seen in the very early 1980s in the Galapagos. There was a series of back-to-back El Ninos, so warm water events. So um, typically they don't happen in succession like that. So you get a cooling period in between, and things that don't like that hot water have time to readjust and get back into their comfort zone. But there were back-to-back El Ninos. So there was a period of time when there was high temperature of water. And since that time, that coral species had not been seen. It was well known. It was abundant at several different dive sites, and it had been looked for extensively in the zones and places where it had been. Like I said, we always start with the last place it was seen. And so scientists had been looking there for obviously, 40 some years, trying to locate this coral species, and it was thought to be extinct. There were conversations on whether it should be declared, which is a lengthy process. But um, the mesophotic divers, the rebreather divers, had been to one of those sites last year, and obviously they were diving deeper. They were at 65 meters, and they were taking video, not thinking to look for this coral. Why would you look for it? It doesn't live there. It's a shallow water coral. So, but they were taking video, and back in the lab, everyone was kind of looking at the video, and someone was like, "Hey, stop. There's there's a Wellington's coral there." And everyone was like, "No, that's impossible." And they looked at the video and looked at it again and said, I'm pretty sure that's the coral that everyone has been looking for." And so we went back, sent them back to the same location, the same site, and they were able to locate and definitively say for sure that it is the Wellington's stony coral. And so the hypothesis is that during these warm water events, the corals spawned, but the larva, instead of settling where they would, right? When corals spawn, the larva is free floating and it can decide where it goes. Instead of settling on the shallow water reefs where it normally would, it went down and found cooler water below the thermocline and said, no, 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 I'm much more comfortable here. This is where I'm going to live from now on. And so we call it a climate change refuge population, something that it ends up in a very unexpected place, but works. So it's an adaptation. So yeah, very pleased
That's
to know that
crazy.
that coral is still with us on this planet.
Yeah. So, is it possible that the coral that we think we already lost to climate change just sits 20 meters further down?
It's possible. I mean, we've now shown. I don't think this coral is the only example of things that have adapted to new and different. It depends how fast it happens too. I mean, the last couple years there have been extreme heat waves in like the Florida Keys, for example, that are continuous and prolonged, that are killing huge swaths of reef. It is possible that they don't have it there. There may not be a thermocline. The Galapagos is, it's more temperate weather to begin with. So it's possible in places like Florida, there isn't a thermocline that they could possibly settle into that would be cooler. I think it's hard to say. There is hope. There is hope that that could be the case, but I think the conditions have to be really specific for that to take place.
I mean, that's the beauty of the scientific method, right? It's like now there can be a hypothesis, and maybe we can rediscover more of these things. Again, a technical question: is a coral an animal or a plant?
A coral is an animal. It's an animal that lives as a colony. And so what you see, those kind of individual branches and polyps and things, it's many animals that codepend on one another to live as a group together. Um, it's sessile, which means it does have that larval stage where it's floating about and it moves, but once it settles, it's there forever. It can't. It's not like anemones can get up and walk away. They tend to stay in one place, but if something
Mm.
bothers them, they can move if they need to. They can pick up their foot and kind of shuffle along. Corals cannot. Once they're settled, they live where they live with all of their buddies as a group.
That's so interesting. Fantastic. We're moving closer to the end. I want to understand now. You've been to the Galapagos, and how many species did you rediscover, and how did you also have to declare any extinct?
Um, Not yet, but we rediscovered the coral. Also, in that same El Nino event from the 1980s, there was a fish, the Galapagos damsel fish, um, which has also not been seen, and again was very abundant. So not seeing it it's a prominent fish. So uh, I mean, I don't want to say it's not there. We took the eDNA. We're waiting for the analysis of. I mean, we took hundreds of samples, so it could get picked up. It has been sequenced, so we know its genetics. If there's a match, we can match it directly to that DNA from that damselfish. Um, it's not looking great. I will say it's not looking good. Again, it's something we should see if it's there. And lots of trained taxonomists doing studies at. I mean, we were at 10 different reef sites where it was once abundantly seen. So we're still jury's still out on that. It's not looking great, but we're still hopeful that maybe the DNA picks it up and we're just not
Mm.
seeing it.
I mean, the Galapagos are known to be one of the most biodiverse places on Earth, if not the most biodiverse, I think. Is it more obvious that something is wrong if you go there than if you go to other places?
That's a good question. I think one of the things about the Galapagos is it is because of its uniqueness, obviously the Darwin connection, all of those things, it is very well studied. So you don't have the problem when you go to, next year we're going to the Tagian Islands in central Sulawesi, which are, I'm finding almost impossible to get to. I mean, it's just it's going to be a trek just to arrive, and there is no infrastructure or um, accommodations or anything. So we have to bring all our gear with us, including, the gases for diving and things. So the fact that the Galapagos have an established research station, they have the Darwin station there that we worked with, and people who've been returning. We were with one scientist from Brown University who has returned for 40 years, every year doing fish surveys. So you can say that it's very well studied. And so if you have a biodiversity baseline, then you know what's not there and what you should be looking for. So I think that's a plus. Is just the amount of survey effort that's already gone in there, allows you to track over time.
Mm. What happens now with all this knowledge that you gather if you rediscover the coral or see that a fish might be gone?
Mm-hmm.
Yeah, what happens to it?
Well, through those assessments, if it becomes obvious that it can't recover to pre El Nino levels on its own, there are a series of interventions. And I know that my colleagues there have already begun investigating the possibility of setting up like an aquarium like aquaculture system there so that they can bring up samples and then propagate them, give them the best possible early childhoods without any kind of predators and with really controlled conditions, and then replant them. And so essentially rewild that coral. One of the projects that I am most in love with is the Reshark project in Raja Ampat, and now it is also expanded to Thailand. And that project, so Raja Ampat used to be just a desert. It was dynamite fished. It was not protected, and the government of Indonesia set up a really robust system of marine protected areas there, really well enforced, really well financed, and it has returned. I mean, it's unparalleled. I finally was able to go dive there, and it's ruined me forever as far as diving because it's just so amazingly gorgeous. But the zebra shark population there became so low that it wasn't able to recover. Basically, there were so few adults, they couldn't find each other to breed. And so while there's a healthy population off the coast of Australia, there was almost no population in Raja Ampat, and they're a keystone species. They're sharks. So for healthy reefs, they need sharks. So zebra sharks are really well known in aquariums. They're very easy to keep. They breed really readily. They seem, in your terminology, happy and content. So a group got together and decided that they would it's also the genetics of those populations are really well tracked. So, there isn't inbreeding or any of those other things.
Mm.
So they got together and they started shipping the eggs that were laid in aquariums to nurseries that were built by local people in Raja Ampat. They're hatched, they're taken care of till they grow to a certain stage in floating pens in the sea, and they're taught how to eat and forage, and they're given trackers so that they can see what's going on. And they did a really great population study to know they only have to release like 300 sharks to recover the entire population for the area. And they're already well underway. I think they're close to 50 releases now. And it's just a really successful, well-managed, well-studied, well-researched, well-peopled project that's doing great things. Really optimistic.
That is so beautiful. And it brings us back to the aquarium almost, huh?
Yeah. And why it's important. Exactly.
Yeah.
No, I should have stared. Maybe I should have led with that. That's why aquariums are important, the Reshark project.
So maybe at the risk of opening a completely new conversation, I heard about this company, I think Colossal something that try to bring back species from DNA samples. If there are samples of this Damsel fish that you mentioned. Could we bring them back up in an aquarium and release them in the Galapagos and hope they survive? Or?
Yeah, so Colossal sponsors the Lost Species Program. They're a
Okay.
part
Yeah.
of that. With that hope, they have a huge reintroduction fund and conservation fund. So while they're doing this amazing biotech work, they're also using what they're creating, the technologies they're creating to bring back some of these extinct species can also be applied to species that are on the brink of extinction right now that have very low populations that need genetic support. Um, so it's amazingly well applied technology that has great promise for conservation applications.
No, interesting. And then one last technical question. What is a cornerstone species? What defines it?
Oh, keystone species.
Keystone.
So
Yeah, sorry.
yeah,
Yeah.
keystone species are the ones that if they're not there, they will cause environmental collapse. Um, so one example is the California sea otter, the northern marine sea otter. They were hunted to extinction for their beautiful pelts back in the 1900s, and they were thought to be extinct here in Monterey. And unfortunately for the bay, so they are voracious eaters of other species, particularly sea urchins, and sea urchins eat kelp. Kelp obviously supports the entire ecosystem here in Monterey Bay. It's where the fish have their babies. It's where all the habitat that hides everything, that everything lives through. It's very important. The kelp is very important. But without the sea otters, the urchins grew out of control and they started eating the kelp. And now we have what's known as kelp barrens. But through the Monterey Bay Aquarium, they've been doing work with sea otters. There was a population that was found. They've been helping to rewild genetic populations of sea otters here for years now. And it's a healthy population, which is now keeping the urchins under control, which are allowing the kelp to grow, which are allowing the fish populations to boom and rebound. So it's that a keystone species is that species that if it's not there, all else fails.
Interesting. So can a fly be a keystone species?
A fly could be a keystone species. Anything can be. Yeah, definitely.
Is this
Definitely.
part of your job to define that, how important an animal is to an ecosystem?
Partially. I mean, part of deciding of those 4,500 species on that lost species list, we can't look for everything. So, how do you target and what is that? And being a keystone species is one criteria of many that could be important and a reason why resources should be put into that.
I learned so much today, Christina, and I know that there's a way how people can also support the work. Do you want to mention how?
Yeah, so as part of that Galapagos study that we just did, as I mentioned, we were testing new technologies. I'm developing a system that hopefully can be used um, by recreational divers worldwide to help support the data gathering for reefs through eDNA. And so we had brand new technology that we were testing out. Of course, you don't know what you don't know. And so after we got done with the Galapagos study, I realized that we needed a couple more pieces of data. And so I've developed a second study that we're going to do right here in Monterey. And it's being funded by the California Biodiversity Institute and also through their channel experiment.com, which is a crowdsourcing network. So if you go to experiment.com, you can read about this study and donate if you want to help support it.
Wonderful. And people, remember, even if you don't care about the environment, if you like to sit behind your computers, the results of these studies directly impact your life. So please, if you can, support the project. Christina, I can't thank you enough for all the insights that you gave to me. I'm now a good step closer to be educated on this wonderful topic. So thank you so much. And I could keep talking to you forever.
I know, it's so fun. And one of these days you'll have to come with on a expedition and come see it firsthand.
I would love that. We have a project in North Sulawesi, so maybe it's not too far to Central Sulawesi, but I would love to.
Awesome. Well, thank you so much, Joel, for having me. I appreciate it.
Likewise. Thank you so much.